Hello, myself Dr. Hridayesh Arya, Department of Zoology, N.R.E.C College, Khurja, and my topic is Canal System in Porifera. As you know, the vital life pulses of a sponge are dependent upon a continuous, uninterrupted flow of water through its porous body. This is essential because they are sessile and have other means of getting food and oxygen or getting rid of waste. Water current is caused by constant beating of flagella of millions of choanocytes. Water enters spongocoel through ostia and exits from osculum. Flow of water is controlled by closing and opening of stomata. Elimination of metabolic waste, chiefly ammonia, also takes place by diffusion through the surface of body. So it is very important to study about the canal system in Porifera. Canal system is a distinguished feature of all sponges as it has a perforation of body surface by numerous apertures for the ingress and egress of water current. Inside body, the water current flows through a certain system of spaces collectively forming the canal system. As you know, the sponges are holozoic and brings various kinds of microscopic organisms as food such as bacteria, diatoms, protozoans and other organic particles with water current inside the body, and the selected food are digested by the choanocyte cells of the flagellated chambers and serve the purpose of nutrition. So the arrangement and complexity of internal canals is divided the canal system into three types: the Ascon type, LEUCON type, and Sycon type. The most simple type of canal system is Asconoid type, and some complex in Syconoid type, and the very complex in LEUCONoid type. The most vital role in the physiology of the sponges is played by water current on which their life depends. All exchanges between sponge body and external medium are maintained by means of this current, and the food and oxygen are brought into the body, and excreta and reproductive body is carried out. So it is continuously done by the beating of flagella of collar cells. I'm speaking about the Ascon type canal system, which is the most simplest type of canal system, having hollow and gastro-like constructions with thin body wall. It is having central large species spongocoel lined by a cell layer of choanocyte. There is an unfolded layer of choanoderm. Canals runs straight through the body wall. Straight entry of water into the spongocoel through the tubular porocyte, and these structures impose very definite size limitations. Thus, the asconoid sponges becomes always small, vase-like, and grows in groups attached to the rocks in shallow seas. You can see here the diagrammatic representation of Asconite type of canal system, in which you can see here the small pores or dermal ostia from where the water enters into the spongocoel. The spongocoel is very bigger and wide, and water goes outside through a narrow chamber called osculum. It is the course of water current in Ascon type of canal system. The incoming water reached into the spongocoel through ostia and goes outside through osculum. Sycon type of canal system is slightly complex than Asconoid type of canal system with thick folded walls, and the foldings of the body wall retains the radial symmetry. Central large spongocoel is not lined by choanocytes, rather complex water passes a twisted route, constriction of choanocyte to the evaginated radial canal, and these structures help to increase the surface area of the choanocyte layer and allow sponges to increase in size and help more water flow efficiently through the body. The syconoid sponges pass through an asconoid stage in their developmental stage, suggesting that they have evolved from some ancestral asconoids. It is the course of water current in Sycon type of canal system. The incoming water reached into the incurrent canal through dermal ostia and goes to radial canals through prosopines. With the help of Apopiles, it goes to the spongocoel and goes outside through osculum. It is the Sycon type of canal system diagrammatic representation. Here you can show the foldings of the body wall with dermal ostia. These are the porocytes. These are the choanocytes cells. It is the radial canal. These are the apopyle openings. And the water goes inside the spongocoel through these dermal ostia and goes outside through the osculum. LEUCON type of canal system is a very complex type of canal system with no spongocoel. Highest degree of folding taking place in this type of canal system. Numerous incurrent canals lead into the small rounded choanocyte chambers necessary to propel more water through the body. Canals are large and branched. These structures help to increase significantly in the surface area and the system seen among the largest size sponges. The complexity of structure of the leuconite canal system helps to increase the most efficiency other than canal system. The leuconoid canal system may have evolved through asconoid and syconoid stages. Course of water canal in LEUCON type canal system. Here you can see the incoming water goes to incurrent canal through the dermal ostia and goes to flagellated chambers through prosopyle, from flagellated chambers to excurrent canal through apopiles or small pores and goes outside through osculum. It is the diagrammatic representation of LEUCON type canal system. Here you can see there is no spongocoel cavity and the osculum is wider in comparison to spongocoel, and the water goes inside through the dermal ostia and incurrent canals. Rhagon type of canal system. In demospongial leuconoid canal system is derived from a larval stage called Rhagon. Spongocoel of Rhagon is surrounded by flagellated chambers opening into the very wide apopiles. A single osculum opens at the top of the spongocoel, and the canal system of Rhagon larva does not occur in any adult stage of sponge. In demospongy LEUCON type canal system is also termed the Rhagon type because of its derivation from Rhagon stage. Comparison between different types. Compare with body wall, which is simple in case of asconoid type, evaginated and with incurrent canals in syconoid type, irregular in leuconite type, and simple in Rhagon type. Mesenchyme is simple and thin, completely transverse by porocyte in case of asconoid type of canal system, thickened and not completely transverse by porocyte in case of syconoid type of canal system, highly elaborated transverse by incurrent canals and subdermal spaces in case of leuconoid space, considerably thickened transverse by incurrent canals and subdermal spaces in case of Rhagon type. Choanocytes are lined the spongocoel in asconite type of canal system, limited to the radial canals in syconite type of canal system, and limited to the flagellated chambers, and the flagellated chambers opens by narrow apopiles in case of leuconoid type of canal system. The limited to the flagellated chambers with open by wide apopiles in Rhagon type of canal system. Spongocoel is spacious in asconite type, spacious in syconoid type, but obliterated or narrow in case of leuconite type of canal system, and somewhat spacious in also Rhagon type of canal system. So you can say it is totally depend on the canal system, which is the exclusive feature of the phylum Porifera. So it is really important to learn about the canal system of Porifera. Thank you.